Hi, good evening.
Good afternoon.
Afternoon. All right. My name is Daniel, and I would be your tutor for this course, this biology course, right? Um,
Biology.
is biology, right?
It's advanced.
Oh, I think you're in the wrong room.
Oh, okay. Just now.
I think advanced is room one, if I'm not mistaken.
Okay. All right. Sorry. Thank you.
That's fine. Hi, good evening.
Good evening, sir.
Are you here for biology?
Then who is this, sir?
Are you here for biology class?
Yes, sir.
Okay, good. All right. So my name is Daniel, and I would be your tutor for as long as you have class here. Um, Just some quick questions. Are you in Form 3 or Form 4?
Form 4, sir.
All right, good. So we'll just start with the CSEC syllabus going forward. Um, have you ever done biology before? This is the first time you're doing biology and it was integrated science before? Or what's the extent of your knowledge on the topic? Subject.
I did biology before, sir.
Okay, no problem. All right. So we don't have to stick, we could just jump right into it. So the first thing on the syllabus is uh, So living organisms in the environment, that is section A of the syllabus. I'm gonna pull up some slides here that I have. Yeah, let me know. You can see my screen?
Yes, sir.
All right, perfect. Right. So living organisms in the environment is the first topic that we will look at. And the first objective of this topic is to group living organisms found in a named habitat based on similarities and differences. In other words, and the next objective is to classify organisms into taxonomic groups based on physical similarities. Right? So, and feel free to stop me if you have any questions, right?
Yes, sir.
So living organisms can be classified into groups based on similarities and differences, right? You can classify anything based on similarities and differences. You can classify humans by height. You can classify dogs by the color of their fur. You can classify different insects based on whether they have wings or don't have wings. For example, simple classifications could be made based on observable characteristics, such as number of legs. So if you were given a sample of insects and you see some have 8 legs, some have 6 legs, so you know characteristics of insects is that insects only have 6 legs. So the ones with 8 legs would be maybe a spider, and they would be arthropods. They wouldn't be insects. That's an example, right? Presence of antenna. Some would have antennas, some wouldn't. Some would have hair, some wouldn't. And that's just a few categories that you could look at when um, classifying organisms. Have a lot of others. Living organisms could be divided into large groups called kingdoms. Kingdoms are then split into phyla, phyla into classes, classes into orders, orders into families, families into genera, and genera into species. So before I ask, no, you don't need to know, you don't remember that by heart. That's complicated. But you do need to remember the kingdoms and the species, right? Members of a species exhibit same basic characteristics and can successfully reproduce with members of its own species successfully. So you understand what they mean by that, right?
Yes, sir.
Right. So they're basically saying if two animals could reproduce and make viable offspring, meaning good offspring, they are members of the same species. That's why two dogs would mate and make puppies, but a dog and a wolf, while yes, they have a lot of similarities and they come from similar backgrounds in terms of classifying, a wolf and a dog cannot make pups, right? That makes sense.
Yes, sir.
So, right. Any questions so far?
No, sir.
All right, good. So, like I say, you have to remember the kingdoms. The kingdoms are just the basic overarching names. So, you have the prokaryote kingdom, which is single cell organisms such as bacteria, cyanobacteria, autotrophic, heterotrophic bacteria. Basically, bacteria, amoeba, anything that's a single cell organism would fall under that kingdom. Protoclista is any organisms resembling the ancestors of our plants, so like ancient molds and algae would fall under this category. Then you have fungi, which is fungus. So any um, heterotrophic, non-motile food digested outside the body, any fungus like mushrooms, um, penicillin, all these different fungi that grow the mold on your bread, all fall under the fungi kingdom. And then you have the plant kingdom, autotrophic, meaning that they make their own food, right? So plants make their own food from photosynthesis, and all plants would fall under the plant kingdom, just as all animals are heterotrophic, meaning they need to get their food from another source, whether it be plants or another animal. They are heterotrophic, meaning they get food from another source and they digest food and remove the waste. Right? Any questions?
No, sir.
All right. So this is just a diagram showing you how you could classify the different kingdoms. Um, so this first one, I don't know why this diagram saying archae, but this is prokaryote, right? So these are the single cell organisms like different bacterias. This is, or they split it, never mind. One, two, three, four, five. 1, 2, 3, 4, 5, 6. These two, kingdom bacteria and kingdom archaea, fall under kingdom the prokaryote, right? As you can see here, it have the protista, which is the algae and the different types of algae, different types of protozoa. And you have kingdom fungi, which is the mushrooms, mold, yeast, and then you have plants and animals. Straightforward, right?
Yes, sir.
Right. So the plant kingdom could be divided further into different groups. You have mosses and liverworts. You know, mosses grow on a wet surface and is be slippery. People is slip and fall on moss, but they usually tend to grow on surfaces like this that you see in here, covering the whole surface and not growing higher into a plant. And then you have ferns. Ferns are classified by leaves. These types of leaves that you see in those plants would fall under ferns. And then you have the gymnosperms and the angiosperms, right? So gymnosperms are plants, are seed producing plants. You could also call them conifers. Um, they produce seeds like pine cones. However, that would fall under gymnosperm. Angiosperms, however, are flowering plants. Those are plants that would have a lot of flowers. Flowers to attract insects for pollination and whatnot. They are usually pollinated by a vector, meaning insect, bird, etc. Gymnosperms might be pollinated more by the wind. So the wind will blow their pollen to other plants, and through that, they'll be able to fertilize another plant and reproduce going forward. Right? Any questions?
No, sir.
All right. And another thing in gymnosperms, their seeds tend to just fall off, and the seed will eventually will also um, grow into the ground and eventually grow into a young plant, provided the conditions for germination are met. Right, and they have six basic animal groups as well. You have the invertebrates, you have fish, you have amphibians, reptiles, birds, and mammals. What category of animal do you think you fall under?
Mammals.
You sure it's not a fish?
Yes, sir.
Okay, just making sure. So it could be anything these days. Right. So how to classify these organisms? So based on the picture you're seeing here, what you could look at, what characteristics some of these organisms carry that you could use to kind of separate them.
The number of legs they have, sir.
Yeah, the number of legs, any others? Or that's it?
Some of them have wings.
Right. Good. Another one that you could use for insects especially is the number of segments. So you see in here how this one have a head, a body, a back, a head, a body, a back, and some have all this rigid structure instead. And look, this one have a head, one, two, three segments, and others have this shell that divided into multiple segments rather than the normal one, two, three that you see in most insects. Right, good, good. Right. We just went through the grouping organisms in a named habitat, right? So that's what this was. This was an example of organisms in a named habitat. This is if you're to go in the backyard and you sample and you get all these different insects, you could now categorize these insects, classify these insects based on different characteristics. So you say dragonfly have wings, the ants do have wings, etc. Right? Classify different organisms into taxonomic groups based on physical similarity. Now, you wouldn't watch a fish and say a fish is a reptile, and you wouldn't watch a bird and say a bird is a mammal. This is what we know from since primary school. It ha- with regards to classifying animals. Plants, however, I understand um, people could get mixed up, but it's a matter of just reading and remembering because you have mosses, you have ferns, gymnosperms, and angiosperms, and they all are different, but they have distinct features that make them what they are. Right? So we can move on to objective two. So objective two begins with carrying out a simple ecological study using the most appropriate collecting sample methods. Right? And then after that, we'd have to distinguish between abiotic and biotic factors, niche and habitat, population and community, species and population. And then we have to discuss the impact of abiotic factors on living organisms. Living organisms could also fall under biotic factors. Right? So the first sampling method we will look at is the use of quadrants. You ever used a quadrant before?
No, sir.
Okay, so a quadrant is simply a square with a known dimension. It could be biggish, it could be small. Once it's a known dimension. And what they do in the field, they would carry the square and they would just toss it. And wherever it lands, they would count the different species in the square, what they're looking for. So you see how this one on the ground here, as scientists would now stoop down, I will count how much ants, how much batcha, how much lizard it have in the square, how much each animal, if it's small garden animals to study. And then he will make a note of it. And then he will go to a few other places that are a bit distant from this. They wouldn't show it in the same exact place, but in the same area of study. Toss it a couple more times and see how much species he would get, different species he would get in each area that he researching. And what this does is tell us the species diversity or biodiversity as well as species density. So it will tell you how much of a particular species is found in an area. So if I toss a quadrant outside my yard in three different places and I get 20 ants in one, 10 ants in the next, and 40 ants in another one, that just telling me how much ants, like the amount just telling you how much of that species you find in that area. If I throw a quadrant and I get ants, like 10 ants, 14 batchak, 3 lizard, a snake, that's the diversity in that area because I have different species in the area that the quadrant in, right? So, like it says, a series of squares of a set size that are placed in a habitat of interest, and species in those quadrants are identified and recorded. So they identify and they record the species, and then they would go about um, checking for biodiversity, checking for species diversity. Um, what do you think is the importance of knowing if a area is biodiverse or if an area has a high species density? What do you think would be the importance of knowing these things, these measurements? Hello?
Yes, sir.
Yeah. What would you think would be the significance of knowing the biodiversity or the species density of an area?
To know the different types of animal species and which type of animals they are.
Yeah. Anything else?
To know if it has a balanced ecosystem.
Correct. So you would know um if the ecosystem or if one of the species decline, if you can tell the different feeding patterns by the amount of species it have. Um, in terms of biodiversity, the most diverse as a area is the better because it have more options for feeding in terms of what creatures you would find there. So yeah, it's to understand patterns of how animals interact with each other in that particular area and if the ecosystem, like you say, is functioning properly. Right. So these are some other collection methods. Uh, we didn't go too much into them because at the end of the day, the quadrant is the most significant one that you would be coming into contact with. Are you doing biology in school or you doing it on your own?
In school.
Right. So when you start doing labs on ecology, you would see they would carry you out and they'd give all of these squares to go and toss in the yard when you're doing labs. And you would have to count different species. It might even be animals, it might be different types of grass or whatever it is they ask you to look at. So we have these others. The first one is a potter. Uh, Potter is a very weird thing. It's like you ever see somebody siphon gas from a car, or just put the hose in the can and suck on it until the gas come up, and they'll put the hose and let the gas continue flowing. You ever see something
Yes,
like
sir.
that?
Yes,
Right.
sir.
So a potter works in a similar way where you have a jar, and in one end it have a hose and the other end have a hose going towards the area that you're studying, and you have to pull through the hose coming to you like a siphon and pull what you're trying to sample into the jar to study. Um, I'm gonna see if I can get a picture to show you because this is a funny instrument. And I have to change this. This is it's not Potter, it's Puta. It's double O, not double T. Um, right? You see the guy with the jar,
Yes, sir.
right? So there's what it is. So he put in the straw on the tree. Maybe it have a little crease in the tree in the bark that he trying to get that insect, trying to get a sample of the insects, and he pulling through this side, and it will just deposit the insect into this the suction will pull the insect out and deposit it into this jar so he can take it to study. A very strange instrument. Excuse. Right. Bottles and jars are self-explanatory. In just a bottle and a jar, you pick up your sample, you put it in the bottle, you carry back to study. Same thing with the jar. Uh, you can use those for studying samples, dry samples, of course, like insects or any small creatures outside. Um, you have sieves. So, you ever sift flour before?
Yes, sir.
So, a sieve works in the same way. Is a big square that is just like a sift that is used for flour, and you're taking a sample of dirt or sand, especially. This is work really good if you're trying to study organisms on the beach. You could take the wet sand or the dry sand, toss it in the sieve, and shake the sieve, and it will sift out the sand, and whatever little organisms it have would remain. So, for example, if you go to a beach and you're studying um, whatever organisms exist in the sand, you can take a sample of wet sand from an area, put it in the sieve, and then shake it, and you'll see the little tiny insects, something that is be borrowed. You might get a little hermit crab or something in between. So yeah, sieves just like a sip. That's all you have to remember. Quadrants, we just talked about quadrants. It's a square that you just have to throw and count and record your sample. A belt and a line and belt transect is let's start trying to remember. Is a line is like it laid out across an area to be surveyed, and a quadrant that is put a quadrant in the place in the first. There's a divider. How to explain this way? There's divide a piece of land, into different areas, and they will place the quadrant on each area, and they will follow that pattern going forward to identify the abundance of a species in a overarching area. And a line transect is just a line. Say you standing at point A, and you take a line and you run across the point B. You're going to follow the line after, and anything on that line, around that line, is what you counting and recording. And it depends on what you're looking for. So any insects along that line, any small creatures along that line, you're counting. Mark, release, and recapture. That is when you, also known as catch and release, that's when you're studying a particular species of animal, and you would catch them, you would study them, take your recordings, and you would mark them. For example, if you catch a bird and you're studying the bird, different birds are the same species. You'll capture the bird, take whatever readings you have to take, and then you'll attach a little tag to his foot. I don't know if you ever see something like that in movies or TV. And then they will release the bird after. So now the bird marked, so they know this check that bird already, and they might try to catch back that same bird to see if anything changed from before. Of course, they will release it in the wild. They'll have an artificial environment for the bird, so they can get back the same bird. Any questions on any of these?
No, sir.
Right. And all these sampling tools are used in the study of ecology. Ecology is the study in the field of biology that deals with the relationships of organisms and the surrounding environments. So, abiotic and biotic factors, right? So these are the definitions that you have to take into consideration when studying ecology. See, we have 5 minutes again, so we'll just go through this, and that will be it for this evening, right? We'll continue tomorrow. But in ecology, you have abiotic and biotic factors. Abiotic factors are the non-living part of the ecosystem that shapes the environment. So temperature, light, water, soil, none of these things have life, but they are crucial for a living environment to function. You can't have plants alive without light, without water, without soil. Depending on the temperature of an area, you might have a particular animal, a species of animal, and you wouldn't have it as well. If based on temperature, for example, you wouldn't find a lion in the North Pole, they'll freeze to death, and you wouldn't find a penguin in the Sahara Desert, they will die from dehydration. Biotic factors are the living part of the ecosystem that shapes its environment. So the biotic factors, like I said, would be plants, animals, fish, insect, anything with life, right? The niche of a species is the role that a species has in its environment re- with regards to interactions of abiotic and biotic factors. So basically, the role of a species being, for example, a kobo. The role of a kobo is what? Not to feed on the dead, decaying carcasses. They keep the place clean. It have other animals with different roles. Some would be an apex predator. Some would be um, food, etc. Um, and a habitat is the natural environment of an animal, plant, or organism. So the habitat is where you would find a particular organism living, and that would be the environment that they are most suited to. For example, like I said before, you wouldn't find a penguin in the Sahara Desert, you would find a penguin in the North Pole because the habitat of the North Pole is suited for the lifestyle of a penguin. A community is an association of populations of two or more species within a given ecosystem at a time. So you will have a community. A community would be, um, let me say, a jungle. A jungle would have a community. You'll have populations of two or more species. So you'll have jaguars in the jungle, monkeys, snakes, insects, all coexisting in a community. Whereas a species is a group of organisms with shared characteristics that can reproduce naturally with one another to create a fertile offspring. So you may have different types of tigers in the jungle, but each tiger would be able to um, reproduce with each other. So a white tiger, a normal tiger, a Bengal tiger, they may be able to reproduce with each other because they fall under the same species. A population is a group of organisms with shared characteristics capable of interbreeding in a given area. So similar, a population is a large any questions? Hello?
I find population and community kind of similar, sir.
Yeah, kind of similar. Trying to see if it have a difference here just now. Okay, so a population is all of the organisms of the same species in the same place at the same time, right? And a community is all the populations in the same place at the same time. So all living things. Um a community is different populations. So all the tigers, all the snakes, all the monkeys, same place, same time, right? Whereas a population is just one. So all the tigers is a population. All the monkeys is a population. All the people is a population, right? But community is okay, like in Trinidad.